Hi, good morning. I'm Ron Shaplow, one of the MLC families here. Thank you for attending both in person and online. I'll be moderating this. If you have any questions at all, please type them in the chat line. I will keep monitoring them. And at the end of the sessions, I will ask them. I'll use Google Translate where necessary. You should be able to get live translations on the Google app. Send me a link if you are, or send me a note on the Alliance MLC if you're having trouble getting in, and I will try and connect you. Anyways, without any further ado, I think Christina, you are going first on the session, so I will let you take over. Thank you.
Thank you, Ron. Good morning, everyone. Just before we start, I'm not sure whether the people who have registered online have the right link because I'm not seeing them here, and some people have been asking. So I don't know if there's a way to email it really quickly to everyone who has registered online. But I'll get started. Today I'm gonna talk briefly about the MLC Scientific Symposium we hosted last February. I'll touch upon the MLC patient registry, which I'm sure will then be covered in more detail by our other presenters today. And then how the families can be connected, fundraising priorities for Alliance MLC, and then talk briefly about our the first family conference we want to organize in India. So the symposium took place in Amsterdam end of February. We had clinicians and researchers focusing on MLC from all over the world, from Europe, US, Asia, and Latin America as well. Japan, China, over 25 people came, and 2 MLC families were there coordinating it. And it was really the first time everyone was under the same room, in the same room for a day and a half. There was a lot of information that was exchanged. Everyone left having learned a lot and also having been inspired to continue working on finding a treatment for MLC. Also, the members of our MLC medical consortium, clinical consortium, were there, and it was the first time that they met in person. A few learnings from the symposium were that, and I think that's what most families want to know. Yes, we do have still some way to go to be clinical trial ready, but a treatment can be imaginable, is possible, and it's a matter of time. There were areas of research that were, in terms of focus, that were identified. There was a discussion on gene therapy, and Dr. Bosch is going to elaborate on that today. But there is definitely progress in gene therapy. And then what was really discussed and is really important for the families to know is that the registry has the work has started, and we'll hear from Nicole a little later. In order for research to be able to proceed and get to being ready for clinical trials, we really need to have a patients registry, an international registry, and the natural history studies that will come out of this registry are urgently needed because they will feed the research with key information in order to move forward. So it's really, really important for all of us, the families, as soon as the details of the registry in terms of the rules and the questionnaire we will all be answering. Once everything is set up as a network, international network, we will be receiving information on how to register. So that's really, really important. And then we talked about the next symposium, which would be a symposium that will be for the families. And the most sense, given how many cases are in India, it makes sense for the first symposium to be in India, and that's the next one of the next projects we will be taking on. So to elaborate a little bit on the patient registry, as I mentioned, it's critical to start clinical trials. We, the families, need to register, and that means answering a questionnaire. It is a questionnaire that will be quite lengthy, and it will require all of us to sit down and look back and provide information on how the disease has affected our family members. So it's definitely not going to be a pleasant necessarily process, but it's absolutely necessary. The type of information that is collected, it's information that we have from doctor reports, from MRIs, and then data family. However, it's important to mention that all this data is safe, is protected. And the idea is to create a network registry that will start in Amsterdam and then expand in other areas. And then, of course, as I mentioned, we, the families, will need to participate, but also motivate other families to do the same. And this is really the only way to get to clinical trials. In terms of connecting with families, we have an ambassadors program. We have ambassadors in key countries, India, Turkey, several countries within Europe, US, Canada, Japan, and Australia. We have a Facebook group. It currently has 123 members. It's at this point it's the one place, and this is a private group, but we also have our public Alliance MLC page. But in the private group, I really urge everyone to become a member. You just need to ask to be invited, and we will, of course, accept your invitation. A lot of things are being discussed, questions families have, concerns. We each talk about our experience. In my opinion, it's an invaluable group, and because we see the doctors every few months, and that's important. Our kids go through therapy sessions, but there are a lot of other things that in this lonely process we get to discuss, and we invite everyone to be as active as everyone wants to. We also have a newsletter that we send out every couple of months, and you can register on our website. So I urge you to do that. That also has about 150 members right now. And then we are also on Instagram. We're still getting familiar with Instagram, and it's not as active as the other platforms I mentioned. A fundraising update, obviously, a big part of what Alliance MLC does and will be doing is fundraising for a number of things. For example, I was very proud to see that we were able to raise $15,000 to support the scientific symposium in February. That was completely supported by the families, and this was our first project. And so I want to thank everyone who participated in any level possible. We have applied for a grant, and we'll continue applying to other grants. The Zuckerberg initiative does support organizations like us, and we will know in August if an opportunity for a grant will come our way, which could change what we do significantly in terms of accelerating what we're able to do, especially in the fundraising front. Immediate fundraising goals, the registry to get this in place was a 400,000 euros. Sorry, this number should be euros here. Project with the help of Dr. Van den App and some family donations. We've raised a little over 300, and we have 100,000 to go, which really is part of what we, the families, have to raise. I would say about 15 of that has been raised, so 85 at this point. We will also work with, especially with the families in India, to plan our first family conference in the next 1 to 2 years. As soon as we know certain research needs, including gene therapy, we will also start participating in supporting specific research programs in the future. And then about our first family conference in India, as I mentioned, and what we heard last year at ULF, India does have, at this point, for what we know, the largest number of MLC cases, along with Turkey, another important country for MLC. And so we're in the process of setting up Alliance MLC India. And Dr. Sinha, who presented last year here at ULF, is helping us and is an integral part of all our activities in India, along with 2 other doctors who have had many MLC patients. And so Alliance MLC India will be critical in helping us organize the first family conference there. We also, with the initiative of Dr. Sinha and our ambassadors in India, we will start planning local clinics that will go to MLC spread areas. The idea is to educate the families about MLC, offer them services like a one-stop shop for everything, all the tests, exams, everything that the MLC patients in those countries need to have access to, but also to start educating people, especially in India, in terms of trying to stop spreading this disease within specific communities. So yes, as I said, our current ambassadors in India, there's also about 22 families that are forming a group, and Dr. Sinha will be very important in our activities there. So how to get involved? You can become an ambassador, or we can never have enough ambassadors. You can fundraise for Alliance MLC, and we can talk offline on how you can help us do that. You can connect us with more families in the area where you live. You can provide administrative support to Alliance MLC in your area of expertise, where it's a volunteer-driven organization, and so I would love to get everyone's help. And then, of course, you can also make a donation. So there's different levels of involvement depending on everyone's capacity. And with that, I just want to really say that I do think that if we come together, we can accomplish many things, and we can help research move forward and get to the day when there is a treatment for MLC. Thank you, everyone.
Yeah. So we founded a consortium in 2023, and we chose 7 clinical scientists with expertise in leukodystrophies and with a focus on MLC. So we chose the people depending on their expertise, but also for geographic representation. I'm going to try. Yes, wonderful. So we are located in Amsterdam. Tigiyasha Sinha is located in India. Then Pinar Topologlu in Turkey. Then we have Francesco Nicita in Rome, and Jennifer Bernard in Canada, Ali Fatemi in the USA, and Paulo Scobi de Souza in Brazil. Of course, this is not the whole world, but this is about what we can deal with the time difference. Because we have to, if we meet and it's 7 a.m. in the morning in the USA, it's already in the evening in India. So that's why it's chosen this spread. Our work is supported by Eline Hamilton. She was my PhD student. She's finished, and she did the first natural history study also on MLC. So she wrote a paper on that subject. Then here is Nicole Verzavel. She is currently my PhD student, and she will give a presentation on the registry, how she's doing that. And the mission of the consortium is most of all to work on clinical trial readiness, because MLC is a bit of a neglected disease, and very little clinical information has been systematically collected over time. We did a little bit, but we could do better. Then the question is, what does it take to become ready for clinical trials? So you need information on disease forms, because it's impossible to design a trial if you don't know the disease. For instance, a disease that's fast and that is rapidly progressive requires a different trial from a very slow disease or highly variable disease. So in MLC we have the classic disease, then we have relatively benign forms and relatively severe forms, and a remitting disease. And the registry is meant to document it also. Hopefully over time Nicole will help us collect all this information. Then also information on disease determinants, like does it matter which gene is mutated, because we have right now 4 genes for MLC, MLC1, TPRC5B, aquaporin-4 and gliocam. And clinical details, do they matter? Does it matter how big your head is? I always have the impression that matters a bit. MRI details, environmental factors, we have no idea about that. And then most of all, we also need to work as a consortium on validated outcome measures. So if you have a trial, what's going to be the entry criterion, and how do you measure then that a therapy has worked or not worked? And then this is also very important. We need to know what patient numbers there are and where they are. So it's wonderful if everybody is in the registry, so that if there's a trial, if there's a therapy emerging, we can look for eligible candidates, and we know where they are, so that they can be contacted, that the patient could be eligible. So this all can be achieved by patient registry, as is going to be explained by Nicole. And this registry is going to be governed by a combination of the MLC clinical consortium and the patient advocates. So most of all, Alliance MLC. This is what we think about. This is an example. So we have a board with all consortium members. Then we have a management team, and we have a scientific advisory board, patient advocates. We have importantly, a data access committee. So it's not that just everybody can get data out, but there needs to be a deciding body that evaluates the project and says this is a good idea. So somebody like a basic researcher in Baltimore can ask for a specific data set, and then the person gets the data set. So nobody gets just access to the raw data. People can get data sets, which is different. And then we can have all sorts of different projects. As you can see, a natural history study, phenotypic variation, impact of disease, and health technology related studies. The current consortium efforts are mostly monthly meetings. We meet online, we meet for 1.5 hours, and we are almost finished with making I hope that I can, yeah. we almost finished with making a customized clinical questionnaire to document the disease course, and the first draft is finished. And after this meeting, I'm planning to send it to Christina for the input of families on the questions that we ask. Perhaps we overlook certain aspects, or you feel like we overstress certain aspects. And what we have also done, and Nicole is going to explain that, an IDELPhi. Oh, I'm not good at this. An IDELPhi on assessment skills and questionnaires. And then this is my final slide. This is my actually a little bit old picture of my team in Amsterdam. So I have to thank a lot of patients and families for everything that they were willing to do for me. I have to thank the patient advocates, but most of all Alliance MLC, but also ELA and the ULF. This is our center, and numerous collaborators, funding agency and supporting departments. Thank you so much. Anybody with questions?
Is there a goal number for the amount of families for the registry to be able to move toward clinical trials? Like, how many are we looking for?
Well, I would say as many as possible. For instance, the Vanishing White Matter registry, we almost have 500 patients in, and then you can also choose. And what is, for instance, an important thing is for gene therapy, you cannot do a placebo control. You cannot just inject something in the brain of somebody or inject something else, so you will know who's going to be treated. And that means that you need historical control information, and then you need matching of patients. So they should be, for instance, I think the same gene and not another gene, and they should be matched according to disease severity and disease duration in order to be able to see whether after 2 years or after 5 years the therapy has worked. And so the more patients, the better.
But given MLC and its sort of rarity, is there a minimum number to be able to begin?
Well, I would say we work with what we can get. I would say a nice minimum would be 100. And but I think actually, especially with the help of Dr. Topologlu, and she's going to give a talk today, and the help of Dr. Sinha, we might be able to get higher numbers, and the higher the better. And then we also urgently need to start, because you need follow-up information. You need the information from now, but also like we started the Vanishing White Matter registry in 2004, so that registry is 20 years. And then you can really say something about the disease course. And we are just, for MLC, we're just starting. So that's a lot of work on the shoulders of Nicole. But she is doing her very best, and I sometimes feel bad, because I also make the MLC consortium people work. And then, we say what they need to do and which skills they need to assess and things like that. So it's work. It's work for everybody. Another question.
And I guess just sort of similar. Similar to a follow-up, I guess, to that.
Yeah.
Is then once you have the amount that you need to start, how soon can we start to see clinical trials start?
Oh, that does not depend on the registry. That depends on the therapy. I was just saying to Marla that MLC is, I find it a difficult disease. It's not the worst disease, it's a rather slow disease typically. And that makes that you need to be very careful what you're doing, because like if the patient dies because of your therapy, you have done bad damage. And the mechanism is about iron and water homeostasis in the brain, which is also something that is so very important that the brain has many systems to do that. And if one system fails, then the other kicks in, and that makes it also complicated for therapy. So, like simply a drug, I don't envision soon. And Dr. Bosch will speak about gene therapy. And I was very happy when I heard, I hope she still wants that, that she also needs to go to do big animals in order to be for it to be safe. Like if you have cancer, like a pons glioma. Typically patients with a pons glioma die within 2 years. Then you can take risks for a therapy. But if you have MLC and patients typically live for decades, then the risk that you're going to accept is lower. And so I think this is very commendable that Dr. Bosch is going to take the safe route. And then I think, you know, I'm a sort of simple type of person. I think that we just have to make the steps, and we can ask ourselves, how long is the road? The road is long. But let's just start to make steps. So we make the registry, and then hopefully as many people as possible can fill it in. Then we can start data collecting. I think the work that Dr. Bosch is doing also takes quite some time, because doing big animals for gene therapy is very expensive and also takes much time. So, but we are moving, and that's different from in the past when there was nothing. We are now moving. That's at least something. Anybody else? No, then I think that Nicole, you are the next speaker.
Yes.
And Nicole will talk about the registry. I think I give you the microphone. That's okay, right?
Yes. Hello everyone, can everyone understand me so clearly? Yes. Yes? All right. Well, thank you for your attendance. Before I start, I would like to thank everyone for their attendance, and it's a great honor and opportunity for me standing here, for which I'm very grateful, and I'm trying my very best here. I'm Nicole Verzavel. I'm from the Amsterdam Leukodystrophy Center, Amsterdam UMC, and I'm a PhD student there. And my project will be about the setup of the International MLC Network Registry. And I will be walking you through some introductions, definitions, and I will be talking about the progress and current activities, but also about our future and next steps. Well, first of all, what is a network registry? We have a little bit touched that, we've touched it a little bit, but I would like to define the definition a little bit more. A network, according to the Cambridge Dictionary, is a large system consisting of many similar parts that are connected together to allow movement or communication between or along the parts, or between the parts and the control center. And a registry is a secure database containing all relevant demographic, genetic, and disease-specific information of those affected by the disease. And these two definitions give a clear picture of what a network registry entails, as it are multiple secure databases which are connected to each other, which allows communication and movement. So this not only allows local studies within participating centers, but also more global, overarching, bigger studies. So now you know a little bit more about a network registry, but why is it important? Why do we need a network registry? As previous talks already told, it's very important for research. For instance, the natural history studies are very important as they gain more insight into the natural progression of the disease. Furthermore, disease determinants, for instance, predictors for the disease. You could think of blood biomarkers, for instance, blood biomarkers are very important. You could look into that a little bit more, but also you could set up necessary guideline components, which are essential for clinicians who are unfamiliar with the disease in order to effectively diagnose and treat those affected with MLC. So we've already touched a little bit upon why natural history studies are so important, but they are also important in the next steps, as they are very important for drug development. in clinical trials, because trial setup is determined by natural history studies, as natural history studies gain more insight into the natural course of the disease, and trials without knowing anything about the disease course are not possible. You have to know when something progresses or something stabilizes in order to know if treatment is effective. Furthermore, natural history studies are also very important for regulatory decision making. Regulatory decision making are considerations regarding the potentially risky and expensive treatments. So when a treatment is on the market, you want to weigh the benefits, the risk treatment, side effects versus the severity of the disease. And they make a balance between the cost and the disease severity. And the disease severity can be described in those natural history studies. So again, natural history studies are very important. And my conclusion is, a registry is very important. But why do we need this for MLC? As all of you already know, MLC is a rare disease, and the population is sparsely divided around the world. So no center can manage a registry on this scale, and we cannot just ask for you to fly across the world for just one physical examination. That's not feasible, and we don't want it for you. So our aim is to start in Amsterdam with a registry and then replicate this around the globe in order to set up our network registry. So our overall aim is to establish an international network registry aimed at facilitating research on MLC, but also preparing for clinical trial readiness and providing a platform for these regulatory decisions making. But setting up a network registry is not something you do in one day. So we divided those in smaller steps. So currently we're determining and selecting important outcome scales for the MLC registry using a modified Delphi procedure. And when I talked to Mario first about setting up the registry, she said, oh, we're going to do a Delphi procedure. And I wrote it down and I looked it up and I thought you probably won't know what it is as well, so I put it on the slides. But a Delphi procedure is a process used to arrive at group opinion or decision by surveying a panel of experts. So it uses multiple rounds, which allows interaction between the experts, but also ends when a consensus is reached. So it's actually a beautiful procedure in order to reach consensus. So for establishing our endpoints for the registry, we need different aspects. We need our experts, and those are the consortium of the MLC. Those are the consortium of the MLC. Then we did a literature review on clinical endpoints, because what endpoints are they going to be voting on? So we looked at other leukodystrophies, such as MLD, vanishing white matter, and of course MLC. And we looked at clinical trials or trials that not have yet been started, but had their clinical endpoints defined in their action plan. So we gathered that, and then the results of our literature review, we had 390 references. That's a lot. Also, we had 196 possible clinical outcome measures, which is also a lot, and it's too much to be voting on. So what we did, we made a pre-selection of measures to vote on, and during 7 rounds, we came down to 36 measures for the Delphi procedure. So these 36 measures are not the endpoints yet, but these are the points the consortium is going to be voting on. And then we made also a division between the physical assessments, so these physical examinations, the clinical skills, which can be filled out by clinicians based on written information or just seeing the patient once, and then questionnaires, which can be filled out by either the patients or proxies, families, you. So we did that. But now we have our experts, the members who are going to be voting, we have our endpoints to be voting on, and how are these experts going to do the voting? We want them to make an informed decision, so we made some considerations. For all these 36 endpoints, we made a list of these properties in order to give them more insight into the measures. So we looked at the age validation. This is the age range the test has been validated for. As you're looking into natural history studies, you want to follow children when they are young, but also when they are teenagers and when they grow older adults. You want to have tests that measure those efficiently. Furthermore, we looked at the internal consistency. These are the items on a test that measure the same domain. These, you would think that a test always measures the same domain, but that's not always the case. So it's important to be aware of that. Then the retest reliability, also very important. This is the difference in scores between different time points. So if I test someone now and in 3 months later, and nothing changes, the disease don't progresses, it stays the same, you want to reflect that in the scores as well. You don't want to see a difference in the scores. Then the inter-rater reliability, which is also important as we're talking about an international collaboration, is the difference in scores between different raters. Furthermore, the sensitivity to change, the responsiveness, is the detection of progression. It looks almost the same as a retest reliability, but it is something different. This is when I test someone now and the disease progresses and something actually changes. You want to reflect it in the scores as well at a different time point. Then, also very important, the language availability and the validation. This is the feasibility of use in multiple countries because you want to use the native language. And then ease of administration, it's the most important one, the duration of administration, as we don't want to keep you in the waiting rooms for hours, and we don't want you to fill out questionnaires for hours and hours and hours. Otherwise, you would just don't want to fill them out. So very important considerations. So currently, we are conducting a Delphi procedure to select clinical outcome measures for the MLC registry. And I can proudly say that our first round of the Delphi procedure is already starting, and it will end this weekend. So a small reminder for the consortium members to fill out the voting session. Then we would like to engage with stakeholders such as patient advocates and regulatory bodies in order to ensure that we don't miss anything, that we have all our information. But also if we can skip something to make things shorter, that would also be very important. Then we would like to build the first registry in Amsterdam and replicate this in other participating countries. And then, of course, the most important one is recruitment of patients and families for the registry. And this sentence may sound very cheesy, as the registry's success depends on active participation from patients and families, so you. But it's true. A registry without any participants or any family members is just an empty database. So, setting up an international network registry is a big project, and big projects tend to face a lot of challenges, and you have to have a lot of considerations. And one of our main challenges will be sharing medical data is vital, but protecting your information is crucial. So, balance requires pseudonymization and strict access control. So no one has access to all data without any authorization, but also informed consent is also a big part of this. Furthermore, we are accounting for cultural differences and healthcare disparities. You not only see these between countries, but also within one country you can see this. And an example for this, for instance, is eating utensils. Not every culture uses them. Some use only their hands, but some use chopsticks and others use spoons, knives and forks. So we're already aware of those cultural differences. And this is an example of one. Furthermore, this is also very important. We want to ensure comprehensive data capture while minimizing the burden on families, on you. For what I've said before, we're striving towards essential outcome measures and minimizing the quantity and duration of data collection requirements. We don't want to have you fill out questionnaires for hours and hours and hours. So this is one of my last slides. I would like to introduce the MLC consortium members, but they were already introduced. For Amsterdam, the Netherlands, we have Mario van der Knaap. For India, we have Giyosha Sinha. For Istanbul, Turkey, we have Pinar Tobeloglu. For Rome, Italy, Francesco Nisita. For Montreal, Canada, we have Geneviève Bernard. For Baltimore, the United States, we have Ali Fatemi. And for Sao Paulo, Brazil, we have Paulo Scobi de Souza. So these are our consortium members or experts who are currently engaging in the Delphi procedure. Then, also I would like to introduce Aline Hamilton. She is supporting the consortium, and yes, this is me. I'm also supporting the consortium as I'm standing here working on the network registry. And then I've been talking, I think, for a while now, but these are my take-home messages. The main point is that the establishment of an international MLC Network Registry is imperative for advancing understanding, treatment, and care for those affected by MLC. And again, my cheesy line, the registry's success depends on active participation from you. And with that in mind, I put a QR code on the slide. Unfortunately, yesterday I received an email that my trial, apparently I had a free trial, it has ended. Yes, sorry for that. I like the idea, but yesterday I received an email that my trial has ended. So I put a QR code on here in order, if you want to receive more information, you could put down your email address and your name, and then when the registry is ready or live, I will keep you informed of that, or send you to the next participating center, the nearest to you. So I have to find something for that. If you want to talk to me, I will put your address down and I will notify you when the registry is ready. So with that, I would like to thank you for listening to me, and that was it. Are there any questions?
I guess when do you expect to be able to send that note to us? Because I'll get on it as soon as, just so long. My question was just around when should we expect that notification that the registry is ready to be filled out, because we're raring to go.
Oh, that's great news. Currently, we're in our first Delphi process, so we're still voting on the endpoints. And that depends on how long that takes, because then when the endpoints are clearly defined, we can build our registry. Building the registry, I'm not sure how long it takes. I think we can start in October. I'm looking at Mario, October, November. So probably at the end of this year, or maybe first in 2025, around then, we hope.
And perhaps we publish, we announce it at the Alliance MLC website, I guess. That might be a good thing, so that it's public when everybody can start.
Yes.
And we also have a register email address, an email address that's specifically only for the registry.
Yes.
So that's also something, but we can add that when we publish it online at Alliance MLC. I'm going to hire a research data manager, an RDM person, in order to set up the registry. I want to do it well from the beginning, because you know, the Vanishing registry is old, and that means that there's certainly some deficiencies that we now have to fix, and that made me think that we really should do it well from the beginning, you know, with adequate privacy control, and how to create a platform in which we can prepare data sets that are going to be shared with researchers, you know, all those things. Yeah.
Thank you.
No problem.
And perhaps I can say something extra about the network registry, because you think why a network registry, but the thing is, thank you. That I think it works better, so that you know your site, the closest site, and that the doctors there are responsible for their own registries. They can also do their own studies. It's only that the registries are exactly the same, which makes that the data can also be shared between the registries. And so you can do smaller studies, you can do bigger studies, because we have seen that with the, for instance, MLD initiative. That's one registry for many European countries, and then the doctors are all very busy and not filling it in so well. And I thought if I make everybody responsible for their own registry, then that might help with the motivation. Yeah, they can also do their own studies, which I think is very important.
Yes.
Any other questions? Any online?
No,
Okay.
you're good.
No?
Thank you.
All right.
Thank you.
Thank you.
So next up, we'll welcome Dr. Bosch, who's going to be talking about gene therapy for MLC.
Can you hear me? Yeah? Okay, good morning. It's a real pleasure to be here. I'd like to thank the organizers for inviting me and giving me the opportunity to explain our results to the families. As you know, MLC is a life-threatening disease. And since we don't know the mechanism by which the disease develops, it's really difficult to hypothesize a drug by which we can cure the disease. So there's only palliative treatment. And in this case, maybe providing the correct gene that is missing or is deleted or is mutated in the disease by gene therapy can be, at least for the moment, the only treatment for MLC. And there are also other evidences that tells us that maybe it would be feasible to treat MLC with gene therapy. And these are the remitting phenotypes that you can find, for example, for example, in MLC-B patients, or even in some MLC1, a few MLC1 patients that have been found with remitting phenotype. Because in fact, these patients have a very small amount of the MLC protein, which is enough to achieve this remitting phenotype. So this is an historic slide of gene therapy. And as you can see, the papers about gene therapy started to be published in the decades of the 80s and progressively increased until the beginning of this century that stabilized. And if we look at the clinical trials of gene therapy that we're promoting during this type, there is a direct correlation. So this means that gene therapy is really a translatable strategy. And it has had ups and downs, but looking at the proof trials in this historical slide, we can see that in the last 5 years, those have been continuously increasing. So I would say that it's getting real. Gene therapy, it's getting real. And based on that, we propose our preclinical strategy using adeno-associated vectors to provide a piece of DNA to the patient, and that this DNA could enter the sick cells and correct them. We need some kind of vehicle. And for that, scientists mainly use viral vectors, which are derived from viruses. Viruses are microorganisms that had evolved through many, many years to be able to efficiently infect the host cells, the patient cells, and to use their machinery to produce their own proteins. So this is something that we cannot do in a more efficient way in our lab than by using these viruses. So we took advantage of this evolution and modified these vectors to translate them into therapeutic vectors. And the advantage of these viral vectors is that with a single dose, we can achieve long-term expression. And this is very important, especially if we want to reach the brain of the patients, because you know that there is a protection of the brain called blood-brain barrier, that if you have to cross it, sometimes it's very difficult, even for traditional drugs. And breaking the blood-brain barrier very often, every week, for example, this is something that we don't want to do to our patients. So we need to find a way to cross this barrier just once, for example, and achieve a stable long-term expression. And among all the viruses that have been tried, we're using the adeno-associated viral vectors that are also used for many other diseases. And the advantage of these vectors is that they can efficiently infect different tissues and transduce the cells even if they are quiescent or also if they are dividing. And quiescent cells are very often in the brain, like neurons or even astrocytes. They are quiescent, and this is necessary because we want to express the therapeutic protein in these cells. And the vector, usually these kind of vectors remain episomal, and this increases the safety compared to other vectors. And in addition, we also modified this vector, so deleted all the genes, all the viral genes, and in its place, we add our therapeutic construct. So they are less immunogenic than other viruses. And so far, there are already 6 AV treatments that have been approved. Some for very severe disease, for example, for spinal muscular atrophy, which is a very severe disease, and there are already near 4,000 patients that have been treated worldwide with very, very encouraging results. But other diseases that are less severe, like hemophilias, for example, had been already approved, or even a disease for a kind of blindness. So it's not that it's only for specific, very severe diseases, but other less severe diseases have been treated. Anyway, this is an example of one AV vector that we manipulate in the lab. And those are the two genes that this vector contains. It's a very small vector. We delete these genes, and in its place, we add the therapeutic gene. And this therapeutic gene is surrounded by regulatory sequences. So in that case, we can modulate when and where and how we express the therapeutic protein. So for example, to treat MLC, we know that MLC1, it's a gene that it's expressed in astrocytes. So what we do is we use a promoter that is specific for astrocytes. So in that case, we can express MLC1 just in the astrocytes. Okay? If you treat a patient with a conventional drug, you provide the drug through circulation, for example, this drug will go to all the cells. Doesn't matter if that cells needs or doesn't need the drug, but it will get it. And sometimes you can have secondary effects due to this drug it's reaching a cell that doesn't need it. So in that case, we can dissect or we can localize the expression of the protein just in the right place. So here we use just a reporter gene with a green fluorescent protein to highlight where our vector can be expressed. And we administer this vector in the cerebellum. We wanted to transduce the cerebellum because the knockout MLC1 animal, that is one of the models that we use for this assay, it was said that it was an animal that mimicked the very early stages of the disease. Okay, so it only has histological pathology in the gray matter of the, in the white matter of the cerebellum. So we transduce the whole cerebellum. We make sure that the colocalization of GFP matched exactly with the expression of MLC1, and we decided to use this vector for our experiments. So instead of GFP, now we cloned, we add the therapeutic construct. And the first experiment we did, in fact, it had 3 different parts. We treated the knockout animal, which at that time we thought the only feature of MLC was the histopathology, the backwards present in the white matter of the cerebellum by three different times. First, as a preventive approach, before the disease onset. Okay, the disease onset is around 4 months. Then another experiment with it, we treat it as an early therapeutic. 1 month after the show up of the backwards. And finally, a late therapeutic 5 months after that. So very much later of the disease onset. I must say that a mouse only lives around 24 months, okay? So in all the cases, in all the 3 cases, we saw the same thing. We corrected the vacuoles that were present in the non-treated animals. Doesn't matter if we treated the animals before or after the disease onset. And we did achieve expression of MLC in the Berman glia, in the cerebellum, and this was enough to, for glial CAM and CLC2, those are 2 other proteins that interact with MLC1. And these proteins are correctly produced in the cell, but they are not located in the appropriate place if MLC1 is not present. So this is what we see here. In the knockout non-treated animal, no MLC, no glial CAM, no CLC2. But in the treated animal, we see exactly the same as in the wild type. So those results encourage us very much. We also look at the ultrastructure of the myelin, and we see that it's very similar to the wild type. In fact, we didn't see statistically significant differences at any of the ages that we treated the animals. So this is giving us, or is suggesting us, something that I think is very important for the treatment. We may have a large window for treatment. In neurodegenerative diseases, where the kids have a very severe neurodegeneration, you must treat them as asymptomatic. We heard something about that yesterday for ALD. We must treat the patients as asymptomatic. But this data, at least in the mouse model, is saying that may not be the case for MLC. So we may have some more time for treating the patients. So we applied for an orphan drug designation and at the European Medical Agency, and we got it in 2021. But we decided to try to do a more translatable approach. And we know that contrary to the mouse model that has the pathology only in the cerebellum, the patients have pathology in the whole brain. So we decided to target the whole brain. And before, at that time, we planned to use a new vector that was able to cross the blood-brain barrier, contrary to other vectors that have been used before. So to cross the blood-brain barrier, we need to inject a higher dose of the vector. And some of the patients that have been treated for spinal muscular atrophy are treated with a huge dose. And we know from the non-human primate studies and the human studies that this can be or can have some severe adverse events. So we decided to treat our animals with this dose, which is safe in non-human primates and in humans. And this is what we did. We treat these animals intravenously at that time, and we treated them at a late therapeutic stage. So when they were 10 months old. Remember that the disease onset is at 4 months. And in the meantime, we have been testing these animals for different behavioral studies. And we found, contrary to what it was previously reported, that they really had some locomotor impairment, at least in late stages of their lives. And the other thing we evaluated was the magnetic resonance imaging, because this is one of the tests that it's doing in the patients. In fact, the idea was to assess for in vivo indicators of the disease that could tell us that we were improving the outcome of these animals. And we also look at the long-term expression. We treated the animals at 10 months, and we sacrificed them 1 year later. Remember that these animals are dying at around 24 months, okay? So at the very end of their lives. And this is what we obtained. I mean, those are two types of behavioral tests that we did. I don't have time to get into the details, but I mean, I can do it in the questions if you want. But in dark blue, we have the knockout animals, and in gray, the wild type animals. And in light blue, we have the knockout treated animals. So at 8 months of age, we see that they have already a discriminatory behavior between the knockouts and the wild type. And we treated the animals at 8 months of age. And from that time of treatment, you can see that the treated animals that behave as the dark blue at 8 months start behaving as the gray ones from the treatment time. Okay, for this test, where we analyze different parameters, and for this other one. This test is used to assess for coordination and motor function. And it's a test that is used for animals that have cerebellar ataxia, for example. And this other one here is more used for a broad spectrum of neurodegenerative diseases. So by combining these two tests, we reach a neuro score that gives us an idea if these animals are ameliorating or not. And in fact, what we see is that these are the non-treated animals, the wild types, and starting at the time of treatment, the knockout animals behave very much as the wild types. And while the other ones, they are getting worse and worse over time. So, and the other test we did, it was the magnetic resonance image. And these are the typical MRI images. And in these images, we did see some hyperintensity in the cerebellum of the knockout animals, but it was really difficult to see. So we decided to change the colors in the MRI in order to see a higher contrast. And this is what appeared when we did all these changes in the color. This is the knockout animal, and this is the wild type animal. We do see this hyperintensity in the cerebellum, but we also see a lot of hyperintensity in other areas of the brain. So despite these animals don't have vacuoles in the cortex, in other structures of the brain, they do have some water. And we did other tests using the MRI. And for example, for the mean diffusivity, that it's the addition of axial and radial, what we see is that it's not that different for axial diffusivity, but for radial diffusivity, which indicates abnormalities in the myelin, it's telling us that there are also very strong differences between the knockout and the wild type in the whole cerebrum, not only in the cerebellum. And the most important thing is that when we look at the treated animals, which is the row in between, they are much more closer to the wild type than to the animals that were treated, the knockout animals. And we quantified all the structures, and I'm not going to get into the details of here, but you see that where we had statistically significant differences between the knockout and the wild type, now they are corrected by the treatment. As we expected, we found also that the vacuoles had disappeared from the cerebellar white matter. And when we look at the expression of MLC in the treated animals, we see that MLC is present in the Berman glia, for example, in the end feet of the astrocytes around the vessels. Those are the normal areas where we expect to find MLC. The expression is also detected in all the areas, and the amount of protein is also very high in these areas. So these are the results we have. So we found a couple of more, a couple of indicators, like the locomotor deficits or the interstitial and intramyelinic edema in the mouse model that could help us to follow the progression of the disease, and this is new. By using this vector as a proof of principle that is able to cross the blood-brain barrier, we achieve long-term correction of the cerebral hyperintensities, and we rescue the motor dysfunction. This is obtained by a single administration when the animals are already adults. So meaning that, or confirming that, I would say suggesting that we may have a wide time window for treatment. So this is not the end. This is just the beginning. We still have a long road to follow. So I told you that we achieve very high amounts of the protein with this system. So we probably don't need that much. No, the remitting phenotype patients tell us that we don't need to achieve wild type levels. And this means that we can lower the dose that we are using, and this would be also safer for the patients, cheaper maybe if we need less virus. So we need to find which is the minimal efficient dose that is enough to achieve the correction of the phenotype. And for this are very important the data with the patients, because finding more patients with a remitting phenotype will tell us which is the threshold that we need to achieve for the correction. This is very important. We are also characterizing other MLC features that these animals may have. For example, if they are anxious, or there are changes in their mood, or for example, epileptic susceptibility to mild traumatic brain injury or fevers, for example, and evaluate if we can correct also this by gene therapy. So we also need to find the appropriate vector. This vector works very well in the mouse, but it's not the same in humans. I mean, we know now that this vector doesn't cross the blood-brain barrier when administered intravenously in the non-human primate. But it transduces the brain if injected into the CSF. So we need to evaluate if are we going to use this vector, or maybe, I mean, there are hundreds of vectors, new vectors that are being developed just now. So we need to find which one would be the best. And we are not alone doing this, because there are many other researchers interested in many other diseases that are trying to find the appropriate vector. So we may take advantage of their studies before we choose our vector. And then we will have to do the biodistribution studies to select also the minimal efficient dose in the bigger animals, and of course the toxicity studies, because we need to start a trial, a very safe trial, before we can treat these patients. So this is the end. I like to thank the people working on this, especially Alejandro, who is a very talented and motivated PhD student, and Raul Esteves. This is 8 years collaboration with Raul and have been very productive. And Ella, who had funded all this work, and Virginia Nunes, who passed away a few weeks ago, but was also a very great collaborator of us. And I'll take questions.
Do we have any questions in the audience here? We have some online, if not.
Hi, thank you very much for your great presentation.
Thank you.
And of course, we all share your reservations and about the safety, but I think it's extremely promising. So very, really great news. In these studies, you focused on MLC1 as a gene, and of course, that's also the largest patient population with those mutations. But can you say something about perhaps therapy for the other mutated genes that are involved?
Yeah. In fact, we have results. I didn't have time to explain them here, but we did the same experiments with GlialCam. In that case, GlialCam is expressed in astrocytes and also in oligodendrocytes. So we use 2 different vectors, one with GFAP promoter and the other one with MVP promoter for the oligodendrocytes. And we did them separately, and we combined them also. We are still finishing to analyze the data, but it looked like separately didn't get enough to completely rescue all the vacuoles. What we don't know, because GlialCam animals have a worse phenotype, they have more vacuoles. So maybe we need more time. At that time, we only look at the 3 months period. Maybe we need some more time before we can do this. But for the AV, intravenous AV, we're just starting this study. So GlialCam is on the road. Yeah.
Thank you.
Okay, we have a question from online. One of the audience members mentioned they read your manuscript with Dr. Esteves in 2021 about the promise of gene therapy, and they were wondering if you could give details about the positives of using it for MLC and some of the positive results that you found.
I have a microphone. Well, yeah, I mean, I guess this is a little bit what I explained here. No, we continue. When we wrote that paper, we only had the first results, no, but then we improve. I think we improve because we have a phenotype, a behavioral phenotype of the mice, and this gives us much more strength. In fact, when after the first paper, we got in contact with some spin-off companies that were interested in investing in our project, but at the end, they always said the same. I mean, the animals don't have a phenotype. Correcting the pathology, the histopathology is not enough. So I believe now we have it. No?
Yeah, thank you so much for your great talk. And I agree with you. One point is that I always say vacuoles in myelin is reversible, essentially, because you can also see it in other diseases. And we discussed this also in February, that I doubt that the myelin vacuoles are everything about in the disease in MLC. So I share your happiness about that you find also a motor, but perhaps also a behavioral phenotype, that you have something to measure that it's clinically important. Because if the vacuoles go away and the patient doesn't improve, then we still have nothing. And may I ask you one other question? Of course, there's a big difference between mice and men, and so what they share is they have cerebellar white matter, and the mouse does not have cerebral white matter. And if you look at the human brain, that is really a big chunk of fatty material. That's the white matter, you know. It's and I'm sometimes worried about the possibility to penetrate the white matter sufficiently. Like I'm now involved in an antisense oligonucleotide trial in which really white matter cells need to be reached, and I doubt it's happening. So what do you think about gene therapy and then this big chunk of fatty material that needs to be reached?
Of course the mouse brain is completely different from the human, but for the moment it's the model we have. I think in the case of MLC, where the target cells are astrocytes, and astrocytes are part of the blood-brain barrier. No, the feet of the astrocytes are surrounding the blood. So I think this is why we have such a nice results, because we reached the brain through the blood vessels. So I think this would be the most efficient way to reach the white matter through the circulation and get into this end feet of the astrocytes. So that would be, to my opinion, the best approach. For the first question, the changes, I mean, the motor behavior, I'm not sure if this is really due to the evacuation phenotype. Is it?
I agree with you.
Yeah. So we are correcting it. So we're doing something else than eliminating the vacuoles.
Yeah, that's beautiful.
And we're also working in this traumatic brain injury and seizures and hyperthermia to induce fever to the animals, and we still don't know the results, but it may also work. So, I mean, we're trying to get as much as we can out of this model, and we haven't started with the behavioral tests after the treatment with the glial cam model, but the glial cam is a more robust model. So let's see.
I must say, and I said this, I really like your cautious approach, because it's not the most severe and progressive disease, and so I like your cautious approach.
Thank you. Thank you. Well, we also need all the clinicians in it, of course, because we cannot select the. Yeah, of course, we cannot select the patients. The first patients we more appropriate to treat, because if the first patients work, then we can include all the other ones in the study. But we need to select the best ones. We need to see what we have to measure in the patients in order to see how the disease advances or not. No?
Yeah, perhaps I may add one word of caution. I agree with you that I also think that we have a time frame of years that you can treat MLC patients, like 4, 5, 6 years. I would like not to suggest that if a patient is 20 or 30, that we can still get the neurological signs go away. That's not going to happen, I think.
That we don't know. Yeah. I mean, they may not be the first ones to start, but maybe if we see that it's safe for the other ones, no, for the first ones, maybe it's worth to.
Any other questions? Any online questions? Okay, so thank you. And our next speaker is joining us virtually. We'll be hearing from Dr. Topoglu about the Turkish experience.
The huge time difference between Turkey and Turkey and EU. It's a great time for me. Everybody left the hospital, and I'm talking from a silent and calm room. Today, I will share with you my MLC experience. Most of them are Turkish patients, as you guess. Turkey is like a bridge between Europe and Asia, and so many rare diseases from Europe and Asia are seen. All of them may be seen. And also in the eastern and northern part, we see lots of consanguineous marriages. So many autosomal recessively inherited rare diseases can be seen, and including leukodystrophies and MLC. I work in Istanbul University Faculty of Medicine, and it's a tertiary center, and they follow many leukodystrophy patients since 1989. I started to work here in 2010, and after working with Mario for a few months, my special interest became MLC since 2015. And as you all know, leukodystrophies can be diagnosed with other medical approaches, with a detailed history, detailed physical examination and neurological examination. But when diagnosing leukodystrophies, three things are very important for us. First, the MRI pattern recognition and combining it with typical clinical findings, and then trying to make the exact metabolic and molecular tests, especially the genetic tests, to reach the definite diagnosis. And we, as you see, these are the pathological classification of all leukodystrophies, and we follow most of them, and we see most of them in our country. And MLC became, as I said, the most important one since 2015. And this is a short slide about MLC, and we know that we see macrocephaly in the first year of life, and cognitive decline, cerebellar ataxia, spasticity, and seizures may accompany these findings. Now we know that there are more than 2 genes responsible for the etiology, and they make a defect in brain iron and water homeostasis and cell volume regulation. Turkish patients resemble very much to all other the world, but all the patients that I follow have MLC1 mutation, and 19 of them are in close follow-up, and I usually follow them in outpatient clinic, but sometimes have to hospitalize them. And 5 of them had a sibling, so 10 are siblings. And after the announcement of consortium, I could reach 25 patients, and now I'm following 25 patients. And I'm trying to reach all the patients in my country, and I think there are more than 100. Since 3 years after my interest in MLC, we are following them in a multidisciplinary approach, and we all work with physical therapy and rehabilitation department, orthopedics, and sometimes neurosurgeons a lot about these patients. And the advantage of my department is that this is a child neurology division under the neurology department, so we can follow both children and adult patients from birth to adulthood, and we can also reach the neurology lab, including EEG lab, easily. And since 3 years, we are, as I said, following them with more detailed tests, including some of them here. And we now making them at least once a year to evaluate the general motor and other functions, neurological functions of our patients. Now I will summarize the 19 patients that I closely follow, and in next meetings, maybe I will follow the rest of them as I see more. The mean age of our patients is 9 years, but we also have 1-year-old patient and 30-year-old patient too. And the follow-up time is about 13 years. And 90% of the first symptom is the increased head circumference, the cause of the application to hospital, and also walking difficulty, behavioral problems, and seizures may all be seen like other patients. And unconsciousness or seizures after minor head trauma is also a prominent symptom. And movement disorders, especially spasticity and dystonia, are most of the movement disorder symptoms. And about our patients' cognitive status, 5 could finish primary school and 2 high school, and 1 could finish university. And the dependency differs a lot according to all age and the clinical findings, but 1 is bed dependent, 1 patient, and 3 are really dependent to their parents, and 6 have mildly dependence, are mildly dependent, and most of them can move and do their self-care, and 6 are independent. 3 have incontinence problems, especially urinary incontinence problems, and all patients can be fed orally, but 3 have some aspiration problems, especially to liquids. Now I will share the symptoms, the general symptoms of our patients by giving examples. Ataxia is seen among 90%, about 90% of our patients. The mean onset age is 3.5 years of age, and dysmetria is also, appendicular ataxia is also bilateral seen among them. Okan and Özkan are one of the oldest patients of mine, and they are siblings, and they have been followed since 17 years, and seizures and ataxia are their main symptoms. But after puberty, their psychiatric symptoms became worsened, and agitation and introvert depression are the main symptoms of their psychiatric situation, and that's why they have to use antipsychotic and antidepressive drugs. But they have also seizures, and sometimes because of these drugs, the psychiatric drugs, their seizure threshold can be down, and they have to use more than 1 antiepileptics, but their seizures are in control. Among all our patients, 75% had unconsciousness or seizure after minor head trauma, and about 68% have ongoing seizures. And 7 have the type of, according to type of seizure, 7 have generalized tonic-clonic seizures, and 4 have generalized plus focal motor seizures. The status epilepticus history is relatively rare to other patients in other countries. 2 had status epilepticus, and generally single medication is effective, and we do not have to go to the maximum dose, and levetiracetam and carbamazepine are the most used drugs. I want to remind something here. For example, sometimes they send us patients that when they see any pattern recognition in cysts or megalencephalic patients, and they usually say that these patients can be MLC and have seizures. But among atypical and challenging cases, for example, seizures other than generalized tonic-clonic or focal motor seizures, or any resistant cases, always suspect about other diagnoses rather than MLC. We also follow behavioral problems, especially depression. Self-mutilation is one of the most important symptoms among behavioral problems, and also aggression and autism can be seen. Spasticity is the most important movement disorder. And about 25% of our patients have spasticity. Here I will share Rukiye. She had diagnosis at 9 years, but now she is 27 years old. And she also have, rather than spasticity, she has dystonia, focal seizures. She was very good until 25 years of age, but they had a flood in their village, and after that psychological trauma, we don't know if there was a minor trauma, but head trauma, but she got worsened, and the symptoms got worse, and spasticity increased a lot. So we sent her to baclofen pump. And dystonia is another movement disorder. Hussein is one of the most challenging patients. He had a diagnosis at 3 years of age. Now he is 22 years old, and he is very resistant to anti-dystonia drugs. He used trihexyphenidyl and he also used clonazepam. At first months, he really answered good to clonazepam, but now the symptoms got worsened, and he also has self-mutilation. When you look at dystonia, you can see that about 25% of the patients have dystonia, and sometimes it's difficult to differentiate it from spasticity. So sometimes the treatment differs, so it's really important to diagnose it. And Tolga is one of the best patients. He's 30 years old now, and he was diagnosed as Canavan disease because he was diagnosed before the MLC discovery. It's a benign case, and he had walking difficulty and his megalencephaly, but afterwards he was diagnosed at MLC1. We can discuss with the geneticist that he has heterozygous for 2 mutations in MLC1 gene, but he is now a computer engineer, and he only has horizontal nystagmus at left gaze, but nothing in neurological examination. The last medical status of our patients is as follows. 11 of them are taking medications, and 13 of them are having physical therapy, and about half of them are using orthopedic orthosis, and one had orthopedic surgery due to severe spasticity, and one has ongoing botulinum toxin injections. And since we are a tertiary center, and after the announcement of the consortium, they send many patients with megalencephaly and the cystic lesions on MRI. And but sometimes most of them have other leukodystrophies, and they were the pre-diagnosis doesn't confirm MLC afterwards. In May, we made a nice family meeting in Turkey. The attendance was not very high. About 19 patients attended, but some were from Germany, some were from France, and it was really good that we all shared our experiences, and we all asked many questions could ask many questions in detail. And after that, patients became to reach me more easily, and now we are planning to repeat it in September, the second one. I really want to thank Mario and all consortium members and colleagues for being the part of the consortium, and for sharing my experience with you, for the all ULF members, and thank you.
Do we have any questions from the audience? Any online questions? Okay. So with that, we'll welcome the next session, which is the family experience. Oh, sorry, yes, yes.
Thank you so much, Pinar, for the beautiful overview. And I think you nicely stressed another problem, that quite some patients have behavioral issues and mood issues. Because we, as neurologists, are typically talking about spasticity ataxia, but quite some patients have also, what I said, sometimes psychiatric features, but mood issues, autism. So thank you for showing that.
Thank you. It's really challenging after puberty. My experience is like that. Until 13 or 14 years, they are okay to deal with, but afterwards they get worse, I think.
Well, actually, that's in my experience also. I've one older MLC patient, and he is really very aggressive. And he is in his 40s. And that's something that I hadn't seen. And he has a psychosis. So that's something I hadn't seen coming.
Mm-hmm. Thank you.
Okay, so I'd like to welcome our families to come up and talk about your experiences. Does the Sai family want to start?
Thank you. Hello, everyone. I'm Stephen Sai, and this is my wife, Jaciria. And our son, Tyler, is 6 years old. He was here a little earlier. He's in the children's room. Tyler was diagnosed at the age of 4. We noticed when he was younger, he obviously had some of the physical symptoms that we all are experiencing, but he walked at about 18 months, right? Was when he started, a little bit later. And we thought we chalked it up to just a little clumsy and uncoordinated. But when he was around 2 or 3, we started noticing some differences, right? Like he wasn't keeping up with his peers and in terms of his motor skills. And so, we started to really ask questions and delve deeper, and ultimately he was diagnosed after an MRI at Nemours Hospital. We're in Delaware, so Nemours Children's Hospital. It's not been easy, as you all know, but it's been an adjustment for us. The biggest thing I would say we've had to learn is the balance between overprotecting and letting him have his independence, but knowing that we're constantly on the watch. We're constantly trying to let him be as normal as he wants to be, but knowing that there are certain things he'll never be able to do. So we've tried to really make sure that he has as many opportunities as he wants to take advantage of, but within safety and structure as much as we can provide. There are a lot of things that Tyler, he's got 4 older brothers, so rough housing and trying to keep up, wanting to do sports, all of that good stuff. We encourage as much as he can safely do, but we know that we've got to keep a constant eye on him. He is an amazing little boy. He is super smart. He's so intelligent, so personable. He does so well in school, and he's just he's an electronics type of kid. Like I was just seeing on that computer engineer. I mean, that's Tyler, right? He loves being on computers. He loves playing video games, and thinking about how he approaches his day to day. He's just positive. He's happy. He's a very loving boy. He gets nothing but praise from everyone around him for his behavior and his attitude, his willingness to just try and go above and beyond. But we've taken advantage of where we are. We're blessed to have a children's hospital near us. We are near Philadelphia, where the where the Leukodystrophy Center is at Children's Hospital of Philadelphia. Our education system allows for an IEP, and he gets an aide in school every single day, who is just one of the best people possible. So, you know, living as parents with this, it's we struggled with acceptance. We were lost. We didn't know where to turn. But as we got into this community and really with Ron and Marla and Marjo and just meeting you all, like last year was our first year here, and just knowing that we have somewhere to go where we can talk to people and connect with other families, it meant the world to us. Like we struggled with even how to tell our families about this because we just didn't know, and we didn't know what questions they would have. We didn't know how to answer questions. And so just being a part of this community and taking advantage of the access to, you know, these folks who are working day to day on our behalf and to find a cure for this has just meant so much to us. So, you know, it's a long journey. We accept that. We're part of it. We're, you know, active with Alliance MLC, and so, you know, we know that we have a long road, but we just try to make Tyler's days every single day as magical and as memorable as possible. And he's taught us so much. He's taught me patience, like you can never imagine. So, do you want to say
I was just gonna say, Tyler has big dreams, and we don't take that away from him, even though, you know, we know what his abilities will be able to in the future. So I encourage as parents to don't take that away from them. Let them have those dreams, you know, because you just never know. Every child is different. And Tyler wears us out. We are exhausted day in, day out, but we just keep going because we don't know when his energy levels will eventually deplete. So, yeah. So.
Any questions from anybody? Well, we're glad to be here, and we look forward to connecting with all of you after the session. So.
Thank you. Thank you.
Yep.
All right. Yes, head on up. So next up, we'll hear from the Jatovic family.
You're excited. Layla loves the spotlight, if you can't tell. So,
all right, who wants the microphone?
I'll take it first if that's okay.
Our last name is pronounced
Jajatovic. Jajatovic. I'm Angela Jajatovic, and this is Layla. Layla is 13 years old. With us also is her big brother Ozzy in the brown t-shirt back there, and my husband Sam. So we're all here. This is our first year here in person. We were on the virtual, we attended virtually for the last couple of years, but this is our first time here in person. So Layla is 13 years old, and I thought I'd talk just briefly about kind of how we found out that she had MLC, kind of how it's progressed, what it's looked like over the years, and some of the things that we've learned from it, and some of the opportunities that it's given us. So similar to the Sy family, so when she was born, it was probably the 2 or 3 month visit to the doctors where they noticed that her head size was larger than average, and so it was through a series of ultrasounds to rule out hydrocephalus, then CT scans, then many MRIs and lumbar punctures. Later, she got her diagnosis from Dr. Renaud at the Mayo Clinic at about 2 years old, 2 and a bit. So that's when we got the MLC1 diagnosis, when she was about 2.5. Layla did walk independently around, it was just before 2 years, so it was a little bit later than kind of her brother who walked around 1 year, or 1 year-ish. You don't remember? No. So she did walk, and then she slowly, it was the clumsiness, she would fall a bit. And so she then moved to a walker at around the age of 7, is when she got a walker, and we got it all, 7 or 8, yeah. And we had a local graffiti artist totally paint up her walker and really like bling it out for her. So she walked with a walker for a couple of years. And then at about 11 years old, so maybe just a couple years ago, Layla got her wheelchair. So she can still go upstairs, but she needs to hold on to a railing. She can't walk kind of unassisted. But the chair is just much easier to get around school and to get around everywhere we need to go. So that's sort of how it's shown itself over time. And I would say, though, that the opportunities, of course, there are challenges that always come with something like this, but we tend to look for the opportunities too. It has also taught us patience. Still working on that, but patience. We see things from a different perspective. You know, for example, how there are many places that still aren't as accessible as they should be. You just don't think about it, right, when you're able-bodied. But when you're, trying to get into a building, trying to go watch your brother bowl, for example, we can't get to where he is because it's up a major set of stairs. So it's given us a little bit of perspective, but it has taught us some really cool opportunities, or it's given us some opportunities like educating others. So what we like to call, so we call handicap parking spots VIP spots. They're not handicap spots, they're VIP spots. So that's where when Layla's with us, we get to park in VIP spots. One of the opportunities that we take is VIP education. So when able-bodied people take up VIP spots, we take the opportunity to educate them, because often you'll see, if they're there, if they've got their handicap parking tag, that's cool. But when we see them not have that, we positively approach them and just suggest that, this is for people who need it. It's not a convenience spot, is what we always have to say, or what I often say. And we just, we've heard all of the excuses. I'm just idling. I'm not really parked here, or I'm just waiting for somebody.
Well, I'm just going in for 5 minutes.
Yeah. I'm just going in for 5 minutes. So we look at the things that it can help us, we've learned from, and we're helping others learn. And Layla's also become a very strong champion of collecting pop tabs. And so she was recently recognized for it in her school. You want to talk about a little bit what you did with the pop tab program?
Well, in Saint Germain, which was my old school, they were collecting pop tabs. But when I moved to a called George McDowell, they weren't collecting. So I started it up again in grade 7. And yeah, so we collected for the recipient of this year, which is Kyrie.
Pop tabs for wheelchairs. Yeah.
Yeah.
So I don't know if there's similar programs. So we're up in Canada, Winnipeg, Manitoba, and she's collecting pop tabs. It's called Pop Tabs for Wheelchairs. So they have to collect 2 million or some crazy number of pop tabs, and then they're able to get enough from the collection to purchase wheelchairs. So every year they have a recipient that gets a wheelchair from all the pop tab collections. So Layla has really taken it upon herself to encourage others and to promote pop tabs for wheelchairs. So I'd say for, yes, it's got challenges, and we are trying to look at it in the most positive way possible, and what it has given us. And it has given us perspective, and it has given us patience and learning. And I think I love what you said about the dreams, because she's full of dreams too, and I think that she'll be able to accomplish all of them.
I have really weird dreams.
Oh, I'm thinking like what you want to be when you grow up. Yeah, we're different kind of dream. She dreams also. And I guess the last kind of fun thing, we've been fortunate through a organization called Variety Club. They do programs and events for kids with disabilities, and they just brought over something called Volt Hockey, and Layla wanted to share her Volt Hockey jersey with everybody. It's something that started apparently in Europe in the 90s, and it's just coming over to North America. It's basically hockey on, electric carts, I guess, and so wooden carts, and everybody with a joystick to drive. And so every kid gets into the same cart, so everybody's on the same playing field, and it's basically like 3-on-3 hockey played on a court, on a gym floor. So Layla started Volt Hockey. She's on a team called Girl Power. It's 3 girls, and they just started their Volt Hockey season this year. So that's another positive. We there's a Sky Center, or what is it, in Manitoba? There's a foundation in Winnipeg that's been really good for us, just giving us opportunities and showing us what's available to help us. And sporting events and other things that's what has helped us. And so just wanted to share that and talk about that. So thank you. Do you want to say something?
Please, if you can, please start collecting pop tabs and tell all of your friends and family to collect as well.
Thank you.
Thank you both for sharing. I'll certainly start collecting all my pop tabs. So next we have a Q&A session. So I'd like to welcome Dr. Vandernap, of course, to come up to the front, as well as our other speakers here. I'll grab another chair. Nicole, yeah, oh my God. Perfect. Okay. Shall I? Yes. So I'll give you guys a mic. Okay, thank you. So to start, if there's any questions in the audience right now, feel free to raise your hand, and I'll bring the mic over. I think it's turned on. It's green.
Thank you to everybody who did already participate to get up there and speak, especially for those family experiences. It can be very difficult to get up and share. So I wanted to thank you all for doing that, for sure. I think that's a very important part. It's also very important to hear from you up there as well, and the ones online. But I do actually have a question. With respect to Aiden, who's here in the back, he does have some dystonia that just would like to get your perspective on treating dystonia versus not treating dystonia, and what you would use if you are willing to share that information. I can share our experience with treating it, and that Aiden's reluctance to now do too much with that.
Okay. Yeah, thank you so much. I think there are multiple options to treat dystonia. And you can use drugs, like dopa medication or something else. You could also use how's it called? Botox in the very active muscles to make them less active. The problem, of course, of the medication are side effects. The problem of Botox is that you have to repeat it. And we sometimes use intrathecal baclofen pumps for dystonia. And I'm a little bit on Aiden's side I was once speaking with a young man, like 16 years old, and I was a young pediatric neurologist, and I saw his dystonia, and I said to the person, do you want me to do something about it, or at least try? And he thought for a sec, and he said, no, because this is how I know my body. And so I'm used to it. And what would you say, Aidan?
I would just say no.
Yeah. Is it also, Aidan, because you know your body and you know how it is now? And if somebody changes it, that's not so nice.
Yes.
Yes, when we did try to treat it, we did Botox, and it was painful, and it didn't last long at all, maybe 1 week.
Oh. Okay.
And then we also tried trihexyphenidyl.
Yeah.
And it was a tapering, increasing dose, and his seizure activity increased.
Yeah.
And it used to be just with head trauma, and then it was just unprovoked.
Okay.
And then we obviously tapered him off of that. And now he's on the levodopa carbidopa.
Okay. And does it help?
It's really hard for us to assess if we think it helps. I think it does. I've been told by one of his neurologists that she believes he's speaking more clearly, that it
Okay.
has helped his muscles in the orally. So
Okay.
hard to know if it helps.
Then I would say stop it, because if it's a small effect that's debatable, then the question is whether it's worth it. So I think that there needs to be a clear effect, and if the speech is more intelligible, that's up to the family. And I think that the doctor sees the person once in so many weeks or months. So I would go for either a clear effect or stop it, because long-term DOPA medication also comes with side effects.
Understood that, yeah.
May I add something?
Yeah, certainly.
I agree with Mario that I think the drugs must be given in severe cases like Hussein I shared with you. He has a really tough dystonia in oromandibular region and neck, and he always suffers from pain because the neck and oromandibular dystonia is too severe and he cannot eat anything. So that's why you can always use botulinum toxin or drugs to resolve it. But for example, in some patients, the dystonia increases with infection, or normally the patient can deal it in normal daily life. But when infection comes, or during a really excitement or other things, for example, the dystonia increases. I think the drugs must be given in severe and like cases, like you're saying. So I think if the patient doesn't want and can deal it with in daily life, they cannot be given.
Yeah, and Pinar, I think we agree that if the patient feels like he has no advantage, that there's no real indication to say, but you have to.
Yes, of course.
Yeah, yeah. Dystonia is difficult to treat. Yeah. So.
Just sort of a similar. With the trihexyphenidyl, we were prescribed that to see if it might help her foot turning in, and they thought the neurologist thought it might be a mix of dystonia and spasticity. It didn't do anything for her, and it was that, tapering up and then tapering down.
But what I would say too, we weren't aware of it, but when we tapered her off, the withdrawal was pretty wicked. Like it was about a week of her head was like, she was just not herself. She was talking really slow. She was saying she didn't know what to do with her head, and she was developing symptoms that it should have treated. It got out of her system in about a week, but from our experience, for others who might be, because we're open to trying different medications to see if they worked or not, but there are withdrawal effects when you do taper them off. That we found it would be just helpful to know.
The problem of doctors is that they are raised to give an answer when there are complaints. So if the family, if a patient says, I have this and this problem, then the doctors are trained to immediately find an answer. And I think what you just said is that you also should then reconsider, is it really helpful? And what's the price I pay? But I think actually that this is also experience that can be added to the Alliance MLC website, because I think other people can learn from it, because sometimes I think families might feel like, but perhaps there is a drug that works, and then it's good to see each other's experiences, to, for instance, know that dystonia is not that easy to treat. Yeah.
Thank you.
Okay, so we have some questions from our virtual attendees. So the first one is that our biggest problem is imbalance. What do you recommend for this?
Imbalance is, you cannot make it go away, but you can do several things. Physical therapy to train your balance is working. Heavier shoes to create more stability, and not very heavy, because then if you have spasticity, then it becomes very difficult to get one foot before the other. But really solid shoes may help. And also, sometimes you just have to adapt, like not ride a bicycle, but ride a tricycle, and then you don't fall over. So I think there are multiple things that can be done. Yeah.
Have you ever tried
Hypnotherapy? Hippotherapy.
Yeah.
I think that patients benefit from it, and they like it a lot. It's also because they love, of course, the horse. They're very motivated, and it again indeed trains the balance. So that's also something. That's a good suggestion. Yeah.
Okay. Our other question is, I have a son, Kuzi, who has MLC1 and has somewhat unresolved spasticity. He had Botox 4 times for this. He is not afraid of operations with anesthesia. What else is not recommended?
Is not recommended or is recommended?
It says not recommended.
I mean, probably recommended.
Well, in Amsterdam we like baclofen, intrathecal baclofen pumps, because they are titratable. So you have a pump placed in the spinal column, filled with baclofen, and then you can, together with the physiatrist and the physical therapist, you can titrate how much you need. So that's really like giving gas in a car. For spasticity and sometimes for dystonia, it works, but it mostly works for the legs, because if the baclofen pump is placed higher, you may get problems with swallowing and speech because it's too high. So it's mostly working for the legs.
Okay. Any other questions from our audience?
This one's for Dr. Bosch. I know you've been doing research with mice. Is there any time or step where it might go to a different animal to see how your research affects different animals?
Well, we don't have other animals which is a model of the disease except for the glial cam knockout animals. So we tried the two of them, and it works in both. For a bigger animal, we will go to non-human primates for sure for toxicity and biodistribution studies. But I don't think they will ask us for other model of the disease in a different species. That would be, I mean, 2 animals is more than enough. Some trials are allowed with just 1 animal. But we have to be sure that our construct is not toxic in humans or in non-human primates, which is the model that we will finally have to go. So those are experiments that need to be done.
And then for Nicole, just clarification on the registry. I know you wanted to start in Amsterdam. So is that for all patients in like Europe to get them loaded into the registry first and then go to different countries, or patients from, say, us in Canada to give you that information to load it up there? So I was just like curious, is it just Europe registry first and then say Asia and then like different countries and continents, or is it all just everybody all at once?
Sorry, great question. I feel like it's a project that you have to do in steps, so you cannot do it at once. I feel like it's the best way to start in Amsterdam, then, for instance, Rome, or then Turkey, and then expand out of Europe.
So when you do the call to families to start filling the registry, is that going to all of us?
I see Mario nodding next to me.
No, the thing is that we haven't figured it really out yet, so I think we don't know. And because I see your question, what would be nice if we have, everything. So Amsterdam is sort of the tryout center to make everything work. And hopefully all the other sites will be up then fast because we worked out the system. And I think our intention is that the patients are registered in the closest by registry because I think that works well. So for you come from Canada, I understand it would be Montreal. Yeah. And but it's not that we have figured out every step of the way completely yet. Yeah.
Any other questions? Oh.
Oh no, I didn't.
Okay.
Do you have one?
I do. Question from the cloud.
Okay, do you foresee any brain surgery for cysts in the brain after the expected treatment is reached?
I think that's a question for you.
Yeah, thank you. No. There's a sort of misunderstanding about the cyst. They are not real cysts, they are pseudocysts. A real cyst has a wall and can grow, but these are just rarefied tissue, and they have no wall. So surgery would be useless. So there are no real cysts. It's my fault. I should have called them pseudocysts. But in a disease name, we found a new disease and we published it in 1995. You are thinking behind your desk about a catchy name for the disease that will work. So and we came up with MLC. And it should be actually MLP then. ML. But I think we leave it. But the cyst cannot be operated. The surgeon would do nothing. And the cyst can also go away because in remitting MLC, they may become smaller and they may also go away. So they are really not real cysts.
Okay, we'll give a moment to see if there's any other questions that arise. You have a question? Perfect.
After the patient has been diagnosed and, they get their initial MRI, do you recommend ongoing MRIs, or is it just like Tyler, he got his MRI, he was diagnosed? Do you recommend more in the future?
I like MRIs. And I would want to be informed. But the thing is that for patients, it typically means sedation or anesthesia. And it's not really that the treatment of the patient is going to be changed. So I would say once in the 5 year is sufficient. Then you sort of keep track on what's happening.
I have a daughter, she's 6 years old. And 4 months ago, she was diagnosed with metachromatic leukodystrophy. My question is, any suggest for her? She start to losing the balance. Her voice start to get low, and I'm searching. We went to Philadelphia with doctor, and I'm searching, try to figure out what can I do with my daughter. I am a single dad, just me and her.
And what did Philadelphia say?
They say that, well, the doctor, Laura.
Laura Adang.
Yes, she's. I was reading and I was searching everything, and they have a treatment in March. Lemmeldi treatment, but I try to figure out if it's dangerous for her or how this works. I don't want to put my daughter in dangers or in some position it's going to be bad for her.
Yeah. Okay, I'm very sorry for you, sir, to hear about your daughter's disease. MLD is a faster disease than the disease we are talking about here. We are talking here about MLC. Lemmeldi is so I go to say it simple, is bone marrow transplantation with gene therapy combined. And so that is that the patient's own blood stem cells are taken out and they are cured. So there is gene therapy done on the own blood stem cells, and then the patient is treated with their own cured stem cells, and that helps if you are early. So if you hesitate and wait too long, then it's not going to help anymore. So you need to do that early. And I would like to say something. What I completely understand is if you're, and that's also for MLC, if your child is diagnosed with a brain disease, that's a shock. And then you want to search in the world, where's the best expert and where can I find the best treatment? So I understand your search. And but there are a few leukodystrophy centers around the world that are really excellent, and the center in Philadelphia is one of them. So you will find in the world similar experts to Dr. Adang, but not better experts. So you are in one of the top centers. And the advice that Dr. Adang gives you is definitely going to be a good advice, because she is one of the experts. And I would also say, if you found yourself an expert, then I would stop the search, because what the problem of doctors is, they all say a little bit something else. And that's confusing. And so if you find yourself a good expert, just stick with the person and follow, and don't wait too long for MLD.
Okay. Thank you. I'm going to stay with her.
You stay with her. Good.
Is that the same with most leukodystrophies, that you need to act sooner than later, or are certain leukodystrophies with treatment later, 18 years later, 22 years later, help?
The problem of the brain in general is that the brain is not very good in repair. So if somebody has a car accident and brain damage, then there is a period of, let's say, 6 months of partial recovery, and then that's it. Then the remaining damage is going to stay, even if you would put in stem cells or whatever. And so the brain, unlike the skin, for instance, if you have a cut, the skin will heal again, and the gap will be closed. If you have brain damage, the gap will be the same after 10 years, 20 years. The brain doesn't do what the skin does. And that's the biggest reason why you need to be, if there's something to be done, then that's in the early stages. For MLD, the period that you can do something, and also for ALD, is rather short. I think for MLC, and I agree with that with Dr. Bosch, it's much longer. It's a couple of years, I think.
Okay.
Yeah. But I know, of course, and you know that I know what your girls have. I think right now there's not really treatment.
No, there's not. I don't even know of anybody else that has it. Everybody else that I know has passed away.
Really?
So, and they're young. Yeah, Emily Rose passed away this past January.
Okay.
At 11.
Yeah. What you could do is, because
my kids are 22 and 18.
Yeah. I know that Dr. Wolf in Amsterdam knows several patients.
Yeah, and I've not been, all I know is from the very beginning when they were the beginning of it with you,
Okay.
of the first 7, and then I heard there were 19, and that's all I ever knew.
Could you please email me? Then I
will.
forward your email to Dr. Wolf, because, you know, I cannot do everything, so I don't work on this disease anymore, but
Right.
but Dr. Wolf does, and I'm pretty sure she has more patients. And you know, what the good MLC is just as rare as the disease of your girls. And I when speaking to the MLC people, it's nice to have a community, right? And to have something like a Facebook page where you can place your questions and meet people that have a similar experience. So that's a sort of support system.
Exactly. We're working toward that. We have a foundation now,
Okay,
and we're
good.
trying to get things out there. It's just hard when you don't have a group to reach out to. You know, if we had that community, like these folks have been fortunate enough to gather together.
Yes,
it
but
would
they
be a
also
lot better.
they also started it.
Yes.
I think you start, for instance, you start with a Facebook page and you invite anybody with the same thing to contact you. I think it will grow, and perhaps you can ask the MLC group how they did that.
Yes.
Christina Maruno would be very good
Yeah, yeah.
for her. 'Cause
Yeah.
Christina, the one that spoke at the very top of today's session, organized it for the MLC families, and she's been amazing. I'm sure she'd be able to give you some good advice.
Yeah.
on
That
how to
would be
start one. Yeah.
very welcomed information.
She did a really nice job getting this together.
Yeah, yeah, I think so too.
Also involved with I'm on the board, so if you want to chat while we're here.
Absolutely. Thank you.
Okay, any other questions? Any online? Okay. Well then, this concludes our session for today. So a big thank you for all our speakers online and in attendance. And thank you to all of you for coming by. For MLC families, we might have a little get-together before dinner later today. If there's any interest, you can come chat with Marla in the back there. And thank you, everyone.